Alrighty. Hello, everyone. My name is Chamithi Karunanayake. I am a doctoral student here at Miami in Department of Chemistry and Biochemistry. Um, Today, I'll be presenting my work with Dr. Rick Page in designing a spectrum of folding sensors for the detection of misfolded proteins by HSV70. Before moving on, I would give you a brief introduction on what I do. Before that, have you ever tried to make a paper crane? If you have, it's kind of tedious and you have to follow the instructions step by step and you cannot skip ahead, you cannot miss a step. Protein cells also do the same with proteins. I like to call it protein origami. So this entire process, we call it as protein quality control. And in a cell, the genetic material contains a message and it will be converted into RNA. Then now RNA has the message, and that message will be read by ribosomes, and then it will make the polypeptide chain by adding one amino acid at a time. Once you have a straight polypeptide chain, it needs to be folded because it's important for a protein to function properly. It needs to have the proper 3D structure. So this 3D structure, we call it the native structure as given in here. And sometimes protein do not always lead to this native structure. So it might have a intermediate folding state, or it can be misfolded, or it can be aggregated. Going back to the paper crane analogy, if you messed up one step while making the paper crane, it might not be properly folded. You might have to squash it and throw it in the garbage. Cells are the same. If the protein is misfolded, it can be chewed up by this machinery called proteasome and recycle the single amino acids back to making the polypeptide chains. Or else, what you can do with the paper crane is you can just unfold it entirely, get the initial flat sheet, and then try to refold it back. So cells also try to do that. They use the misfolded protein, try to get a folded intermediate, and move it back to properly folded native structure. This is conducted by proteins called chaperones. So I study chaperones, specifically HSP70s, and why this is important is, like I was telling here, if protein get misfolded, they can lead into this aggregates. You are really familiar with aggregates because if you heat up milk, there's a curd that forms on top of in the top layer of milk. That's sort of an aggregate. So this happens in the cells, and these can lead into deadly diseases like Alzheimer's, Huntington disease, and Parkinson's. That's why studying protein quality control systems of a cell is really important. Moving back, so I studied the chaperones and basically HSP70s. HSP70s need help from other proteins. One major other player is CHIP protein, which is C-terminal HSP70 interacting protein. Sometimes it needs more accessory proteins like E2 and ubiquitin as well. So there's a huge decision on the hand of HSP70 chip complex that we call as triage decision, where it decides whether this protein is going to be refolded or whether it's going to tag by these smaller proteins called ubiquitin and go to the proteasome on degradation. So I study this triage mechanism, and I hypothesize that different intermediate state and the folding of these intermediate states drives this triage mechanism decision. With that in mind, we decided to make a spectrum of variants that has different folded states at room temperature, some of which are 10% folded, some of which are 90% folded to 100% folded. And we call them folding sensor TPRs or FSTPRs. overall methodology, we scavenge through the literature to find a starting point, and we found CTPR, consensus TPR molecules, and we modified them based on the help of the literature data. And also we did extensive computational modeling using Rosetta software suite. And then we implemented whatever we found through the computational modeling in real life in a lab bench. And we decided to do what kind of stabilizing mutations we have to do, what kind of destabilizing mutations we have to do. And then we got the bacterial cells to make the proteins for us. But bacterial cells make a lot of proteins, so we need to purify these proteins to get our desired proteins. Once I have one particular protein, I conducted CD spectroscopy and then biolayer interferometry. And I still need to process to this step to conduct the Western blots. So to summarize all the experimental data from CD, I got what is the folded fraction of each of the variants. For example, at 25 degrees Celsius, the one that's given in purplish green color, it's about 75% folded. And then using BLI assays, I could decide what is the affinity between HSC70s and the client variant. And usually these are protein-protein interactions, so it's about micromolar affinity, all of which actually agreed to. To summarize all of them, to this point, I would calculate them as like three different variants. So one is 75% folded, one is about 50% folded, and the other one is about 10% folded. So that's where I'm at, and actually the Hilga content and the melting temperatures are correlated with the approximate fraction folding. And with that, these are my references. I would like to thank my advisor and my research committee and previous and current members of PH Lab, and also the wonderful undergrads. And thank you for all the funding agencies as well. With that, I will take any questions you have. Thank you.